Hey guys, Dr. Cummings here from PLNU. We're talking about this article from Frontiers in Microbiology from Bots et al. 2017. What I want to do in this video is just show you how information and or data can be organized in a table. Tables can look busier and more full than a graph, but often they're a little simpler and easier to follow. So if we scroll down to the first table here was our first figure that we talked about in the last video. But here let's talk about table 1. What the authors have done is they've taken a lot of information that would be otherwise in the text in a narrative form that could be tricky to keep track of, and they've organized it visually using rows and columns. So columns are the up and down columns, and the rows are left to right. Okay, so you always want to look at your table description or legend, any footnotes, and tables often have footnotes in order to keep the busyness of the table itself somewhat simple. And then you want to look at the headings of, in this case, each of your columns. And our rows are really set based on the first column, and that's pretty common also. So here we have table 1, summary of the complete genomes of 4 multi-drug resistance plasmids captured from the Tijuana River estuary. Our first column is the name of the plasmid and its accession number. So we could look up the accession number and actually get the entire sequence if we wanted to work with it. The capture method, because apparently in this paper there were a couple different capture methods, endogenous versus exogenous. The size of the plasmid, the incompatibility group, and if that's something you're not familiar with, you're going to have to do a little research and do some reading, figure out what that refers to. The plasmid copy number, how many copies of the plasmid are found per cell of bacteria carrying it. And then this antibiotic resistance genes column really highlights what the authors felt was possibly most important in this whole paper, because the whole paper is all about these resistance plasmids being found out in nature in a wetland as opposed to in the clinic in an infection. And so they went out of their way to actually list which resistance genes were found on each of these 4 plasmids. There are some footnotes, and so we'll look at the footnotes as we come across them. So if we look at our first column, which I said often sets the meaning of the rows left to right, plasmid accession number A, we come down to the bottom, we see footnote 8, GenBank accession number, okay? PLNU11 is the name of the first plasmid with its accession number. Petrie1611, Petrie131, Petrie2011. And then as we go left to right, we now have rows to match up with our columns, right? If this was an Excel spreadsheet, it would all be on a grid. And each of these entries would be in a particular cell or a box in Excel, right? So PLNU11 was captured by endogenous method, and then there's a note on the host, because if you remember, the endogenous method from this particular paper was when the bacteria were first enriched and grown up, and then individual colonies were picked and screened for plasmids. And so the authors didn't know what species they'd be getting. This particular case, through this endogenous method, it was a Citrobacter frundii strain that they call A-TED-A, and it was captured by the endogenous method. It's a 35,840 base pair plasmid in the INCP6 incompatibility group. Plasmid copy number 14.97 plus or minus 3.13. Our INC group B had a footnote, incompatibility groups determined using plasmid finder version 1.3, Cartoli, etc. PCN plasmid copy number per cell. And they tell you that the data are represented as mean plus or minus 1 standard deviation from the mean. That's a measure of the variability. N equals 3 means there were 3 replicates. So this process that they used, if we go back to the methods, we can see they used quantitative PCR, gave them approximately 15 plus or minus 3 copies per cell, and they did it 3 times. Okay, so there's a little bit of variability there, but not horrible. 15 plus or minus 3 seems pretty good. And then which resistance genes were found on this particular plasmid? TET-A and R, full P, Quack-E, Delta, DFR-A10, and BLA-WDC1. As you get more familiar with the literature on resistance genes, these will actually start to make sense to you. If you're new to this, you may want to go look those up. Find out, well, what is TET-A and R? What's a full P gene? What's this quacky delta or DFRA10 or blah WDC1 gene? What are all of these resistance genes that were found all on the same plasmid? Then if we wanted to see what was found on each of the other plasmids and all their various characteristics, all we have to do is work our way across one row at a time. And that's how we use a table. That's true whether the table is sort of summative or summary data like this. If it's raw data, this second table here is still somewhat summary data for the authors, giving the authors a way to visually show the reader what they think is most important. So in this case, antibiotic resistance profiles tells us down here about the abbreviations of E. coli JM109 with 4 environmental plasmids, the 4 plasmids we've been studying, and the 2 original host strains described in this study. So here are the 4 plasmids, pardon me, and then here are 2 brand new strains that they were able to isolate, and they're showing you which antibiotics these bacteria are clinically resistant to. And then they talk about this decreased susceptibility. And so what's the difference between clinically resistant versus decreased susceptibility? Footnotes are going to tell you that, right? You're going to have to go into the footnotes and find out that clinically resistant means it reached a certain level of resistance that is established in the clinic for if these were plasmids found in pathogens. Decreased susceptibility indicates, in this case, that the plasmid made the bacteria more resistant or less susceptible to the antibiotic, but not so resistant that they reached that threshold, that break point, clinically speaking. And so again, if this is the information you're after, rather than trying to find it in text in a narrative, the authors have summarized it all here, so in a glance, you can search through and find. Every table is different, and every table is just going to take some time. You're just going to, the one thing they all have in common is they tend to have columns and rows, and you're just going to have to look at each table and orient yourself, take the time to orient yourself, and spend some time, just like with a figure, working your way through it, and don't walk away from that table until you know what the take-home point is. What's the most important thing you need to know from this table before you move on?